What is DNA? It's super important, folks. Think of it like a giant instruction book, a manual for life. This special book is inside every living thing on Earth. That includes plants, animals, tiny microbes, and yes, even you. The full name is deoxyribonucleic acid, which is quite a mouthful. So we can just call it DNA, nice and simple. This amazing book holds all your unique information. It's the blueprint that makes you, well, you. It's what makes each of us special and different. This amazing DNA book contains many, many chapters. These important chapters are called genes, and we have thousands of them. Genes are like individual recipes or instructions within the larger manual. They tell your body exactly how to grow from a tiny cell. They also instruct your body how to work every single day. Genes decide things like the color of your eyes. They play a role in how tall you might be, or if you'll have curly hair. All these detailed instructions are packed into our genes. Now, sometimes there's a tiny mistake in this very important instruction book. Imagine a little typo, a misspelled word in the DNA code. These small changes, sometimes called mutations, can happen for various reasons. Even though the change might be tiny, it can sometimes cause big problems for the body. This tiny typo can unfortunately lead to certain diseases. These are conditions that can make people feel unwell or for their bodies not to work correctly. That's really not what anyone wants to happen. For many, many years, scientists around the world dreamed a big dream. They wondered if it would ever be possible to fix these DNA typos. Could we ever find a way to correct the body's instruction book directly? Imagine being able to go in and carefully erase a mistake in the code, and then being able to write in the correct healthy instruction instead. That kind of ability could change so much for the better. It could offer new hope for treating many conditions. Well, guess what? Science is amazing. Now get ready for this because it's really cool. Scientists have discovered a new tool. It's like a super smart biological machine that can interact with DNA. This tool is called CRISPR, and it's a real game changer, folks. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Wow, that's another long one. But what it is the exciting part? It's causing a huge buzz in the world of science. This is a revolution in biology, a brand new way to work with life's code. Think about using a computer for a moment. You know when you're writing something and you use the find and replace function? You can search for a specific word or phrase in your document. Then, you can tell the computer to replace it with a different word. CRISPR can do something remarkably similar, but with DNA. It's like having a biological search engine and an editor all rolled into one. This makes a very complex idea much easier to understand. It's a powerful comparison for a powerful technology. CRISPR technology has an amazing ability. It can be programmed to find a very specific part of the DNA. Imagine searching through that entire, huge instruction book for one single word, and CRISPR can find it. Then once it finds that precise spot, it can make a change right there. It's like editing the very code that makes us who we are, or how a plant grows, or how a microbe functions. This level of precision was once just a dream for scientists. It's incredibly accurate, like a molecular surgeon. This ability to edit DNA so precisely is incredibly powerful and exciting. Scientists are really thrilled about what this means for the future. It opens up so many possibilities for medicine, for agriculture, and more. It's a whole new way of looking at biology and understanding life itself. We are moving from just reading the book of life to actually being able to rewrite parts of it. This is a new era for biological understanding and potentially for intervention. It's truly a remarkable time for science. So, how does this amazing CRISPR tool actually work its magic inside our cells? It's pretty clever, and it actually comes from nature. Scientists didn't invent it from scratch. They discovered it in bacteria. Bacteria use it as an immune system to fight off viruses. The CRISPR system has two main parts that work together like a perfectly coordinated team. It's this teamwork that makes it so effective. It's a natural system that scientists have now brilliantly adapted for gene editing in other organisms, including us. First, there's a guide molecule. This is usually a piece of RNA, which is like DNA's cousin. Think of this guide RNA as a super smart GPS or a molecular bloodhound. This guide molecule is specially programmed by scientists. It's designed to match and find a very specific sequence, a particular spot in the vastness of the DNA. It zooms along the DNA strand, scanning the code until it finds the exact sequence it's looking for. It's like a molecular detective homing in on the precise clue it needs to find. Once the guide RNA finds that exact correct spot on the DNA, the second part comes into play. This second part is an enzyme, most famously one called Cas9. Think of Cas9 as a pair of tiny, incredibly precise molecular scissors. When the guide RNA molecule says, "This is the place right here," the Cas9 enzyme gets to work. It then makes a clean cut across both strands of the DNA double helix. It's a very neat and targeted snip, not a random hack. After the Cas9 enzyme makes its cut, the DNA is broken at that specific spot. Now, cells don't like having broken DNA. It's dangerous for them. So the cell's natural repair systems immediately kick in to try and fix the break. Scientists can then cleverly trick the cell during this repair process. They can provide a new piece of DNA, a template with the desired changes. The cell then uses this template to repair the break, incorporating the new genetic information. This is how scientists can change a gene, remove a faulty instruction, or even add a brand new one. It's like surgically rewriting a sentence in the book of life. Scientists are already doing some truly incredible things with CRISPR technology. It's moving from the lab into real-world applications. For example, they are using it in medicine to try and cure genetic diseases. Think about a condition like sickle cell anemia, which is caused by a tiny mistake in a single gene. Researchers are conducting clinical trials where they edit a patient's own blood cells to correct this mistake. This is offering tremendous hope for people with diseases that were once untreatable. The potential for new therapies is enormous. It's not just about human medicine either. CRISPR is also making big waves in agriculture and food production. Scientists can use it to edit the genes of plants. They can make crops stronger against nasty pests or help them grow better in tough conditions, like droughts. Imagine tomato plants that are more resistant to diseases, or rice that produces more grains. This could help us grow more food more sustainably. This means helping to feed more people around the world, which is a huge deal for our planet. But as with any powerful new technology, with great power comes great responsibility, right? Changing DNA, the very blueprint of life, raises some big and serious ethical questions. This is especially true when we talk about making changes that can be passed down to future generations. These are called germline edits. What if we make mistakes that have unforeseen consequences down the line? What if this technology is used for things other than treating serious diseases, like trying to make designer babies with specific traits? We must be very careful and thoughtful. So here we are. We have this amazing, revolutionary tool called CRISPR that could change life as we know it. The possibilities for good are truly mind-blowing, from curing diseases to improving our food supply. But the ethical concerns and potential dangers are also very serious and need careful consideration by everyone. This leads us to a big, thought-provoking question for all of humanity. Just because we now change our genes with such precision, and if so, where do we draw the line? It's something we all need to think about and discuss.